My name is Dr. Alicia Campbell. I am out of the United States, as you can probably tell, but I am a researcher, a scientist that works on MED13L syndrome, and today hopefully I'm going to share some of the things that I've worked on, as well as some other research objectives we have currently in the United States. I'll be representing some of the United States-based MED13L Foundation efforts and hope to give an accurate representation of the research and data collection that the foundation has worked to establish. So I'm from Rowan University. It's a medium-sized school outside of Philadelphia in New Jersey, so across the river in New Jersey. I earned my PhD in molecular cell biology and neuroscience, and a major part of my thesis was studying patient-derived fibroblasts models of MED13L syndrome. So one of the milestones which we heard a couple of times so far for our community was the publication of the gene review for MED13L this past April. Gene Reviews is pretty much the gold standard medical resource that doctors and physicians can use when they encounter a genetic condition that they might not be familiar with. So before this, many doctors had maybe never heard of MED13L or didn't know how to recognize it in a patient in a set of symptoms. So now we have this highly visible, one of the stats was more than 10 million users per year, guide written specifically for physicians and clinicians. So having MED13L represented here puts it on the map. And not only that, it gives families and clinicians an actionable guide that we can use. So what does the gene review actually include? The gene review was written using all scientific literature reported patients at the time, which was just over 100 patients. The gene review itself is extremely comprehensive and includes clinical features, genetic findings, and management strategies. It also provides some guidance on basically how we're able to detect MED13L syndrome, which is most likely, as you're familiar with, through exome and genome sequencing, which can identify 90 to 95% of variants. It also covers an overview of common features and breaks down the percentage of reported patients who exhibit a certain symptom. As I mentioned yesterday, if you weren't here, it is important to understand that this does not yet include caregiver or parent-reported information and is only based on information in studies, peer-reviewed studies from scientists. I also mentioned that in the future, hopefully, we will be able to include and update. We can update as many times as we want and however often we want to include parent or clinician-reported symptoms to potentially change the percentages of individuals with certain sets of symptoms. This also includes genetic counseling resources because most MED13L variants arise de novo, so that means that neither the mom nor the dad carries a mutation in MED13L, but that it arose randomly or spontaneously. This resource provides information about potential future pregnancies and risk factors, also when to seek counseling from a genetic counselor. It also explains the terms that we've thrown around a few times called mosaicism or germline mosaicism. So this would be in the cases where MED13L variants themselves were directly inherited, so opposite of the de novo. But most importantly, this is a global resource. It's helped clinicians across the world recognize and better diagnose MED13L based off of symptoms and more consistently than before. One of the most useful things, this is just a snapshot from it, is the recommendation for evaluations. So this covers all body systems, not just the ones that are seen here. This is only a small portion of that table. This provides recommendations for when to seek evaluations and what evaluators themselves can look for when performing evaluations on someone diagnosed with MED13L syndrome. This can help guide therapies such as speech, physical, occupational therapy, sort of providing a roadmap for how families and providers can coordinate care together and potentially catching complications early on and hopefully helping with early intervention. So we've seen this. Katie put up this little graphic earlier. She has cards and pamphlets. This is a picture of the MED13L research roadmap. The foundation has become essentially the central hub for this community. Families, clinicians, researchers helping foster collaboration. So for families, they've provided education, connections, advocacy, and created a network of parents to help navigate clinical care day-to-day, therapies, and school accommodations. For researchers like myself, the foundation has been instrumental in building and supporting biorepositories, which I'll talk about in a future slide. This includes things such as biological samples, which Simon's Foundation talked about earlier, so that is anything that comes from a patient. The foundation also runs, very similar to this, family researcher conferences where scientists have the opportunity to meet parents to make sure that research priorities are aligned. So us researchers can hear from the parents what is most important from them in terms of what we need to research and come up with solutions for. So all of these things are included along this little journey of the roadmap here, where we are making strides to fully understand MED13L, but more importantly, to identify potential therapies that could be of use. So as I mentioned, to truly understand MED13L syndrome, we have to rely heavily on model systems so that we can study MED13L syndrome without directly studying in a person. So in the United States, we have several biorepositories or banks of scientific models and specimens that are accessible to all researchers. These biorepositories were formed mainly with help from the MED13L Foundation, coordinating the connection and collaboration of a bunch of different families. One of the ones specifically was a collaboration of 12 different families that was conducted in collaboration with Dr. Jennifer Bain, her name's up there, at Columbia University, that's in New York City, where she collected punch skin biopsies. So I have a little cartoon of what a punch biopsy looks like. And from this, we're able to isolate skin cells from all of the children. So now we can use them in a variety of settings, testing a variety of things, which is pretty much the basis of my work, so I'll talk about that towards the end. We also have collection of blood samples, which Simon's Searchlight talked about earlier, which includes the plasma, serum, PBMCs, and it's coordinated through a couple of different efforts. Lastly, we also mentioned that there are induced pluripotent stem cell lines that have been established. These are all tested, verified through Simon's Searchlight. We are also using various whole animal models at the University of Iowa, as well as the Jackson Laboratories in Maine. We have several different mice that have been genetically engineered to have the humanized version of MED13L, meaning that it is representative of what we see in a human being. The mice at the University of Iowa use the Cree-Lox system, where we can essentially turn on or off MED13L in specific tissues, which is important for understanding the development of the animal as a whole. Lastly, we have zebrafish, which I have a little diagram of them in the tank. These are a knockdown model of MED13L to study neural crest dysregulation in the context of MED13L. So in terms of what we can do with the mice, I mentioned that we can look at their overall development, which was actually mentioned in a study yesterday that came out of Dr. Chad Gruder's lab, looking at the facial features of the mice. That is a very recent, as of September, that study just came out. But we can also use mice to test their ability to perform specific tasks. So some of these represented here, such as the open field test, indicates the level of anxiety in the animal, or potentially the neurodevelopmental deficits and the animal being able to understand its environment. Or we can look at things that are maybe more physical, such as the rotorod. This is where mice have to hold on to this rotating apparatus for as long as they can, using their upper body and limb strength. So this could mimic potentially what we see in humans with MED13L syndrome, where we maybe have a loss or a lack of muscle tone. We could see this in the mice and then look to see how they're holding on and how long they're able to hold on to this rotating wheel. All of this is needed to establish a baseline for when we eventually want to test therapeutics and drugs down the line. So we have to establish these models first, right off the bat, in order to see if a therapy is going to work in the future. And lastly, we have antibodies that we have worked with a company to design. Previously, there was only one antibody that was commercially available for researchers to use. And the problem with this antibody is that it was only targeted to one section of MED13L. And it didn't take into account if maybe we have a variant where the entire protein, or as Dr. Small was calling it, the entire train. So if the entire train wasn't there, this antibody would not be of any use to us. So we created more antibodies that were targeted to a bunch of different regions within the MED13L protein. So you're probably asking, what's an antibody and why do we need them? So these are important things for researchers who are trying to understand the level or the abundance of MED13L in a cell. So this could tell us if a cell or cells from an individual is producing enough, not any, or maybe even normal levels of the MED13L proteins. The problem is that researchers, we can't just look at cells and see protein, right? We have a method that's called a Western blot, where we can harvest all of the proteins from a cell, from a patient, and then specifically pull out only MED13L. The word I use here is sticky, so that the antibodies that we use are, for lack of better terms, sticky for only MED13L. So we can have thousands of proteins in a solution, and we're only pulling out MED13L. And then we can compare that across maybe a control sample or between two individuals with MED13L to see what their level of MED13L protein looks like. So far I've mentioned samples, model systems, but another major component in progress for MED13L syndrome is the integration of additional data collection platforms. For example, the biggest one on this screen probably is we are working directly with Boston Children's Hospital to connect a natural history study. This is critical because it tracks how MED13L syndrome progresses over time. So a natural history study tells us what's typical for the condition and then provides baseline information that is needed for a successful clinical trial. So this will be the Foundation's greatest effort thus far in terms of what is currently being funded, and it's absolutely critical in understanding MED13L progression and then being able to recommend therapeutics in the future. Without this, we can't recommend therapeutics. We also have aperc Rutgers University. A recent graduate, Kayla Clevenger, just completed a disease concept model specifically for MED13L, capturing not only medical features, but also the day-to-day impacts on families and caregivers, which I'll touch on in the next couple of slides. We're also seeing new opportunities to produce data through Citizens, which is a platform that allows families themselves to share medical records and histories directly with researchers. So this empowers the families to give them access to real-world data that might not otherwise be accessible or available. So we've heard it before, but I'll put another little plug up there. The MED13L Foundation also supports the RareX data collection program, where caregivers can report any and all information. So these are a bunch of surveys. The information is then accessed directly by credible researchers. Keynote, credible. These are researchers that have been verified by RareX, that have an ongoing active project. They have to give the scope of their project. They have to verify that they're with an institution. So it's not just some random person on a Saturday morning Googling data. All of the information is also de-identified, so all of the names are removed and replaced with a unique code number for each of the individuals before sharing with the researcher. This resource is currently only in English, but with the hopes that it will be expanding in the next couple of months or so to include other languages. So I mentioned the disease concept model, and we talked about it very briefly earlier. This was from Kayla Clevenger from Rutgers University. That was just published as part of her thesis for a program she did in genetic counseling. Also familiar with Rutgers because that is in my home state of New Jersey. So this disease concept model essentially took all literature related to MED13L and combined it with interviews that were conducted specifically with caregivers. So ultimately, this gives us insight into what the literature may be missing. So in terms of the entire study, I have some summary statistics here. The average age of the child with MED13L was 10.5 years, and the average age of their diagnosis was 6.69 years. It was also stratified or separated by both gender and certain demographics, including race and ethnicity, which you can see on the screen. So this is a little snapshot of what Katie was showing earlier, but I wanted to zoom in on this. This is probably the most important figure, as Katie said, to come from this study. Sorry it's cut off, but I really wanted so you could see all the bars in there. Is the comparison of caregiver reports to the scientific literature for specific symptoms and symptom sets. So in purple, just to recap, are individuals from the literature, of which there were 103. And then in green are individuals from caregiver interviews, of which there were 16. So this breaks down symptoms into categories, and we can look to see where there's key discrepancies between literature reported and parent or caregiver reported. For example, there was a huge difference between what was reported for behavioral manifestations. This could very much be due to the fact that clinicians are not seeing the day-to-day that parents live, and not reporting this because they don't see it. The parents see this, and therefore they're able to report on this. It could also be that the clinicians are only focused on the more physical aspects or the growth aspects that they're looking at each checkup appointment that they can physically see in an individual. So it makes sense that we're missing out on some data that could be behavioral. So now we can take a bit of a step back or a step into a microscope to look at what I actually work on, which is the molecular basis of MED13L syndrome. This is where we're able to connect what's happening at the genetic and cellular level with the clinical features that we see in the human. So the MED13L gene, as we know, encodes a large protein that's part of the mediator kinase module, or the MKM. And I call it a sort of center for gene expression. It helps turn genes on and off, and is specifically at the right time and in the right tissues. If MED13L isn't functioning properly, that fine-tuned control is disrupted, and downstream pathways don't receive the correct signals. So one of the cellular effects that we have seen specifically is mitochondrial regulation, and the role that another player, cyclin C, plays in this. So when we have mutations in MED13L, what we have seen out of the lab that I am in is that this other player, cyclin C, is released into the cytosol of the cell. So it's normally in the nucleus. And we also see some changes on how the mitochondria function. There's less of a balance in the energy production and more so a stress response, as cyclin C is able to directly cause what we call mitochondrial fission. It's represented by the scissors up there. So these cellular disruptions may underlie clinical features such as low muscle tone, developmental delays, or even speech difficulties if we don't have enough energy. So specifically in the lab that I am in, we published a mechanistic study in 2022 looking at the localization of cyclin C in the case of MED13L syndrome, where we no longer have MED13L to tether cyclin C in the nucleus. So this triggers mitochondrial fragmentation, which means the mitochondria are constantly being pushed towards a dysfunctional state. Instead of working efficiently to provide energy, they become unhealthy and unable to keep up with the high energy demands of cell types such as neurons, heart cells, or muscle. And this was an important breakthrough because it connected MED13L syndrome to a very specific molecular mechanism and gives us a better understanding of potentially why patients experience things such as low muscle tone, tiring easily, developmental delays, or even neurocognitive challenges. So that original work was done in one patient-derived cell line, but we have since expanded, as I mentioned earlier, to include 12 additional cell lines based on those punch biopsies. This allows us to gear our research towards potential new therapeutic strategies, which is what I'm working on currently. I have my favorite diagram that the mitochondria is the powerhouse of the cell. So if we're able to block and or stabilize cyclin C's abnormal release or strengthen the mitochondrial resilience, we may be able to slow or reverse some of the cellular dysfunction. By taking the fibroblasts donated by MED13L families and testing them in the lab, we're able to identify a molecular defect that is likely contributing directly to the patient's symptoms. So having these 12 additional samples is a major step forward because it expands the diversity of what we can study in the lab. And this allows us to map new variants and link them to patient symptom sets. So we can test therapies in multiple genetic backgrounds and build even better models with this. So our current most promising direction at the cellular level is drug repurposing. So this is using drugs that are already approved for other conditions to target MED13L related deficits. For example, I have up here in mitochondrial rescue, there's 3 main things that we can look at. We can look at encouraging cells to make more mitochondria. We can also help potentially get rid of the unhealthy mitochondria that are there. And lastly, we can prevent mitochondria from becoming dysfunctional in the first place. And we know that mitochondria are very important and very abundant in tissues such as neurons in your brain and in your muscle. So we need a lot of mitochondria because we need a lot of energy. So to conclude, MED13L syndrome research has made some major strides in the past few years, from the comprehensive clinical resources like the gene review, to family-driven meetups, to patient samples and animal models being established. We are steadily building the knowledge we need to move forward. And collaboration across families and clinicians is key. So together, we're making MED13L syndrome not just more recognizable, but more treatable. So I also have my social media is genetically _modified 13. My whole shtick with that, or my thing that I went for with that, was that children who have rare diseases, or specifically MED13L syndrome, are not necessarily mutated. There's nothing wrong. They're just genetically modified. They're genetically modified individuals. So, and I also, 13 is my lucky number. So I went with 13 there. I also have I think the QR code leads to TikTok, but my laptop died before I was able to put in Instagram. But I do Instagram, TikTok, which is just a day-to-day of what it's like being a researcher in the age of social media, where I can show you the inside of a lab and show you that a lab is not scary people in white coats and all sterile, but it's a lot of aluminum foil and plastic wrap and cardboard boxes. And I do a lot of explanation on what it looks like to run an experiment. So if you are in the world of social media, feel free to check that out. And thank you guys for allowing me to come all the way across the Atlantic. And thank you to my husband for making the journey with me. But if you have any questions, I can take them.